pleasure to introduce Anna. So she came in 2015, right? Yeah. To my department and give me the chance to tell this little story. So she has an uncle who is collaborating with a colleague, and I think she asked the uncle whether it's possible to do a PhD in Wildau, and then she made contact because she is from this nano field. She made contact to me via the uncle and asked me for a PhD position, and then I said, yeah, I do not have a position, but if you apply at the German Academic Exchange Service, it may be possible. Then she asked me which subject I would support, and since I I asked her what she did before, and then she described, and then I said, yeah, why not continuing with this and applying with this subject, which she will present. And I said, well, I think it's a little bit science fiction what you propose, but I pretty much like science fiction. And then she wrote the fundament of this application for the proposal to the German Academic Exchange Service for a PhD position, which is funding for 3 years, or 3.5 or so. Of course, I read and improved it a little bit, but this was mainly her work, and she got this grant, and it was then a little bit extended, and she got then a position in my department, of course a non-permanent position, on a project, and she applied for some, I should say, huge funds when she was still a PhD student, and when she was still a PhD student, because she finished her PhD this year in January, she already had her own PhD student, which has been employed last year, and now a second one, and I was very happy that she was very successful with her PhD thesis, not only in terms of publications. Actually, I was always joking about Anna, you are my Ukrainian typewriter, because I don't know how many publications
13 right now.
Well, how much?
13.
13. Yeah, 13 international publications. So, but since I'm not allowed, it's not allowed, my university is not awarding PhD titles, so I always collaborate. I briefly explained this yesterday, and I asked my former PhD supervisor at the University of Würzburg, who is meanwhile a good friend to me and also a collaboration partner, whether he would supervise her. And he immediately agreed, and it turned out to be a very successful PhD thesis. We have a ranking system, and in this ranking system she achieved, it's called summa cum laude, that means with highest honors. And there's only a small percentage of people which get the PhD with highest honors. So I'm really proud, and the initial idea for this school came from her and was taken up by Alina and me, and so I think you can be proud that you can present here now, and I'm really happy that I can introduce you. Nevertheless, I will now leave the talk, because I know it, I have heard it so many times, and I have important issues to discuss with Alina, so have fun.
Yeah. Dear Marcus, thank you a lot for the greatest introduction, and I would like to add that it was a very good luck for me to follow the boss who himself had. Guess which honor for his PhD? It's also summa cum laude. So, yeah. So, let us start, because it was quite unexpected for me. Yeah, thank you. So please welcome you to the first Nano talk, which the title is Towards Synergistic Cancer Nanotherapy. And now I would try to speed it up. It brings us to cancer, as cancer is my primary research area, and it's devastating disease, a group of disease which are responsible for almost 20 million new cases each year and 10 millions of deaths every year. Cancer was ranked as a single factor of that prevents the expected life expectancy increase of lifespan of people in 21st century. So cancer is defined commonly as an evolutionary process during which in the cell following the accumulation of the gradual mutations of the progeny of a single cell. And that different accumulation of mutations gave those cells the selective growth advantage and lead to the uncontrolled proliferation, what is the highlight of the hallmark of the cancer. The original tumor tissue comes from the single cells, but this should not be concluded that all cells are homogeneous, as there are different stages of the development of the cancer tissues, and that starts with initiation, so mainly it involves mutation, and however at this point it's defined not as a tumor but as a hyperplasia and dysplasia, during which is accumulation, the proliferation of the cell's progenitor from the initial one is observed, and over time the mutations give rise to the new clonals of the cells, and this evolutionary process gives rise of the overgrowth of the new clones, which are more increasingly aggressive and increasingly malignant. However, the mutation should not be seen as one, as in general from 2 to 8 mutations should be happen in one cell that could steer to the carcinogenesis. However, this number is relative, as for example in some lung and skin cancers this number can be up to hundreds. So how is it happening? In the adult human body, there are a lot of cell population which is commonly proliferate very actively. So we have 10⁻⁵ cell division per day, and it's common. It's our bone marrow, skin, and the other endothelial tissues, and because of this proliferation, there are divisions per day, and per each division there could be the mutation rate, there could be a state that is very low. It's amazingly low, but still, because of this high number of the divisions, it occurs. Of course, in the cells there are multi-step signal pathways which deal with DNA mutation, so this mutation rate would be even less. But however, if someday at least 2 or 8 mutations occur in one cell, it can turn to the cancer cell. One of the most problematic challenge in cancer is the last stage, it's metastasis. when the cell outgrows not only the tissue of origin, but they evolve to spread along the body via blood vessels and lymph. So there are mainly four types of cancer that are defined by the origin of tissue. It's carcinoma from the epithelial cells, sarcoma from connective tissue, leukemia of the bone marrow, and lymphoma of the immune system. And the most spread cancers in Europe are for the women breast, colorectal, lung, cervix, and melanoma, and for the men prostate, lung, colorectal, bladder, and melanoma again. So, what are those advantages of the accumulation of those mutations that give us the cancer cells? It's the intense proliferation for one hand, and from other hand also the disbalance in the cell signaling. So the cancer cells evolve to evade apoptosis. Apoptosis is a special cell death type, which is a programmed cell death type, and it has an increasingly important role in the homeostasis of the organism. So as we remember those 10 in 5 powers, 15 power of proliferating cells. So at some point it could be too much, but the constant cell number in the organism remains via programmed cell death. So and if the cell are not listening to the signal of apoptosis, the cell is not dying, and it's also one of the hallmark of the cancer cells. And yes, yeah, also for the last metastasis and angiogenesis stage of the cardiogenesis. So, over the history, there are a lot of cancer treatment options, and amazingly that the cancer is much older as a human race at all, as it was found that even 76 million years ago, the Tyrannosaurus rex fossils had osteosarcoma, and however, in human history, we have the first tracing back we can to the Egypt in the time of 3000 BC, where there was first explanation of the breast cancer and its surgery. Then later the surgery was developed, but mainly cancer remained known as a fatal. After 16th century, because of the boom of the development of the life sciences, the cancer disease was also studied and defined more clearly. And first of all, of course, the discoveries in microscope stimulated those discoveries. About the first explanation of the origin of the cancer was done by Wildau on the chronic irritation theory that somehow mimics the accumulation of the mutation during cardiogenesis. Then a big breakthrough in cancer treatment was done after invention of X-ray, Röntgen, and investigations that those radiation are toxic to cancer cell, and with approach of fractionation of the radiation, the treatment of cancer became possible. It means that we can irradiate the tumor from different angles and achieve the local dose in the tumor tissue of the radiation. And from 1940s, the chemotherapy was developed. The first agent, it was nitrogen mustard. It's first alkylating agent. It means that it alkylates DNA in the cell. And it was a product of the war. And after that, the people started noticing that it's toxic against cancer cells. However, it has increasingly very high side effects. So it was not that used so long. And however, the strategy of alkylation of DNA that prevents its further synthesis gave a study where the platinum compounds were developed. And such drugs as cisplatin, carboplatin are used nowadays as well. So it's from this group. Then anti-doxorubicin was found. It's antibiotic from the Streptomyces species. And starting from 1980s, the investigation of the different metabolic and signaling pathways in the cancer cells gave an opportunity to distinguish cancer cells from the normal cells and uniquely target cancer cells. As for example, target therapy exploits the higher activity of tyrosine kinases in the cancer cells, and also monoclonal antibodies to the overexpressed receptors on the cancer cells. And the last breakthrough is goes to the immunotherapy, which got also a Nobel Prize for the finding out the mechanism which how cancer cells inhibit immune cells and restoring the capacity of our immune cells to kill cancer cells. And there are also chimeric cells gene therapy, which is developing. So as an example of chemotherapy drug, doxorubicin, so the idea mainly of the chemotherapy is to stop proliferation. Also could be other mechanisms that includes metabolic components. And if we think about how to stop the proliferation of the cell, we can would focus on the DNA and to how to block the DNA synthesis. It's done, for example, with doxorubicin. It's classical intercalator of the DNA. It means it prevents later of the transcription of the single DNA. There are, of course, more cellular effects of the drug that includes also reactive oxygen species production, so extra nuclear effects as well. So, but chemotherapy struggles from the very common side effects, as for example for doxorubicin, it's primarily cardio and nephrotoxicity. So the applied therapeutic dose of the drug is limited by the side effects. And that brings us back to comparison of the cancer cell and normal cells, and a high important goal to find new approaches how to distinguish them and selectively kill only cancer cells. One of such approaches includes reactive oxygen species and redox balance, as cancer cells are known to have increased reactive oxygen species level. They use it in order to increase proliferation, as reactive oxygen species are also the secondary messengers that stimulate growth. However, over some limit of the level in ROS in intracellular environment, they can cause the cell death as they are highly toxic. So this interplay between normal and cancer cell, when a normal cell, the reactive oxygen level is much less in comparison with cancer cell. So if we increase the ROS level, the cancer cells are probably faster to undergo cell death. Here we come to the nanotechnology, as we can combine cancer therapies with nanotechnology. How? So here one more time, what is nano? And if you know the size of soccer ball and the size of the Earth, you can compare how small nanoparticles are. As fullerene C60 is exactly 1 nanometer big. So C60 is a classical example of the nanoparticle and was already mentioned a few times in the talks of our nano nerds. Thank you for that. You make my job easier. But you could find fullerene under different names. So it's also Buckminster fullerene, buckyball. It was named Molecule of the Year in 1991, and it brought the Nobel Prize to 3 scientists who discovered it. It was in 1996. And so C60 is very easy to see how it's built if you again imagine the soccer ball, because the 60 carbon atoms in the C60 molecule are shaped in exactly same structure, and it could be defined as a nano soccer ball. So we would not go deep to the nanotechnology history, but we can follow again the C60 prominent discovery was there. What I want to tell in a few words is that the discovery of C60 was also a very interesting idea and an example how new investigation, new ideas should grow over common skepticism and critics. From 1970s, there was not one, but quite a series of studies which were pointing on the existence of C60. But they were very criticized, and they were published, for example, the first one in Japanese and this one in Russian. So it was not that known internationally. And it was up to 1985 when Harold Kroto was investigating the formation of carbon chains in the astrophysical environment. And he found his way to the laboratory of Richard Smalley through his friend Robert Kroll, who has very special laser evaporation and technique and equipment where they evaporate those carbon chains from the carbon rod with laser. And they were looking for the chains, and they found some very interesting peaks that were, they studied the mass spectrometry, so they calculated the masses, and the masses were corresponding to the 60 and 70 carbon atoms, and they were playing around how to put them together. As the carbon has 4 valencies, it was a tricky task, but they finally come up with the idea that it's soccer ball. And then they published in Nature, and it's the group who received the Nobel Prize finally. So C60 belongs to the carbon-based nanomaterial, and there are others such as carbon nanotubes, graphite, dots, graphene, and thymine. And the most interesting features of this class of nanomaterials are the optical properties, also the very high stability, mechanical and electrical properties. So I would like to focus on the optical properties, which give a rise of phototherapy. Phototherapy, you can guess that we use the light for therapy. There are mainly two types of the phototherapy. It's photothermal therapy and photodynamic therapy. And due to the special feature properties of the carbon nanomaterials, all of them can absorb light, and some of them can transfer the energy of the light to the heat, and the other can transfer directly energy on the oxygen and produce reactive oxygen species. They're again toxic. For example, fullerene, it's very known photosynthesizer, and carbon nanotubes are better in photothermal therapy. And the differences lays in the gap between singlet and base ground state of the electron. So here once more about photodynamic therapy with C60. So when C60 is illuminated with the light, the electron is boosted to the singlet level, from where it comes to the triplet level, and the triplet level is no longer species, and the time is enough to this excited triplet state of C60 to meet the oxygen, and in the present is there two types via which light excited C60 produces different reactive oxygen species inside the cell, and once this level is high, it's enough to induce apoptosis. So the idea of photodynamic therapy and photothermal therapy as well is also the double selectivity, that we target cancer cells with the nanoparticle and with the light. So the cells which are not exposed to the light and have no nanoparticle would not get any treatment. Here's the absorption spectrum of colloidal C60, which has typical 3 ultraviolet bands. And now we see it with the overlapping exposure spectrum of 4 LEDs, light emitting diodes, and which were used to illuminate cells treated with C60. So it's without C60, it's with C60, so the cell viability was decreasing in the presence of C60 with the increased light fluence of the different wavelengths. That was followed with reactive oxygen species production inside the cells after 1 hour and 3 hours. Also, the ATP content is very important to follow the homeostasis of the cell, and we can observe the drop of ATP content inside the cell. With membrane integrity remains the same, it means it points also on the apoptosis. However, the hallmarkers of apoptosis are one of the executive key spaces through 7 that were shown also to increase their activity after treatment of cells with C60 and light. The phosphatidyl translocation is the final step of the apoptosis, and phosphatidylserine is translocated on the surface of plasma membrane of cell surface and facilitate the recognition by phagocytes of the apoptotic cell. That again we see that after treatment of cells with C60 and the light, the phosphatidyl translocation was found in cells. That altogether points on apoptosis. And with that, I want to finish with photodynamic treatment with C60. and to introduce you the other function which you can use for the treatment of cancer with nanoparticles. It's drug delivery, which Hanna Fedorova already mentioned. So I will add a few words to it that why there are so nice applications of nanoparticles. Because it's the size, it's based on the size of the particles. The nano size grants particles the ability to selectively accumulate inside tumor tissue. Why it's happening in that way? First of all, the tumor tissue, special tissue and blood vessels which are present there, they characterized by very big leakage. So as tumor tissue is growing and it stimulates angiogenesis, those blood vessels and tumor tissue are growing very fast, and they have big leakage. And this the difference of the endothelial wall gives an opportunity for the nano-sized particle to go outside of the bloodstream more likely in these areas as compared with a healthy normal blood vessel. So it contributes to the passive targeting of the tumor tissue and also known as enhanced permeability effect of the tumor tissue. Then the other opportunity is also decorate nanoparticle. As was mentioned already, different proteins can be expressed, overexpressed on the cancer cells, and we can attach the specific ligand to this receptor on the nanoparticle, and in this way, a nanoparticle will stay not only in the tumor tissue but also can be endoseptized, so taken in the cell. So in that way, then the nanoparticle can be used to selectively deliver the drug to our target tissue. There are also other aspects. So to get fast and efficiently to tumor tissue is good, but also we need to consider the homeostasis of organism and all biodistribution and pharmacodynamic inside the organism. And here comes in the play the liver and kidney, which deal with the toxic compounds and secrete the compounds out. And the compounds which are up to 10 nanometers are likely to be secreted very fast. So with the liver, it depends also on modification on the cell and the surface of nanoparticle. But here in the middle is the optimal zone of the size and the characteristic of nanoparticle. So we need to meet the demands of the tumor targeting, but also not that fast biodegradability. So with C60, we studied the drug delivery with a common anti-cancer drug doxorubicin, and we developed very fast the non-covalent modification methods where the aromatic molecule of the drug is attached on C60. So here maybe we don't see that good, but maybe here. So mainly the red light, what you see here, it's the microscopy of the cells, it's fluorescence microscopy, and as doxorubicin is very good fluorophore, we can follow its biodistribution inside the cell. In blue, it's a marker for the nucleus, and mainly when we overlap those images, and we see here by violet light, when that results from the red and blue overlapping, that the doxorubicin is located in the nucleus. And these green dots, which are not seen on the previous slide, but we can follow here it now, corresponds to the C60 accumulation inside the cell. So we can clearly follow that the drug is released inside the cell. Even more interesting that the C60 increased the accumulation of the drug. So it's a free drug, and here is two types of nanocomplexes of C60 with doxorubicin, and the difference of fluorescence intensity proves us of those with those two, shows us that doxorubicin content inside cells are increased upon C60 delivery. So the cell viability also was affected by treatment with C60 doxorubicin. That mainly shows that the free is in red and two nanocomplexes are in violet and blue. This strategy was extended to other drugs, and C60 was shown to increase the toxicity of those drugs against tumor cells as well. And here it brings to the one very great option which nanoparticles grants us, it's multimodality. So as you can guess now, as C60 itself can be used for photodynamic therapy, so it can be toxic compound from one hand, and from other hand it can be a carrier for a drug, so we can combine two therapies together. And C60 is an example from my side, but there are much more different multimodal combinations that can be achieved with nanoparticles. And this design on the molecular level, that we can allow to the nanoparticle different agents, gives us an opportunity to combine even more than two therapies together. So the idea of the synergistic multimodality is that when we have, for example, two therapies, that we get not a sum of their action, but much more, and we achieve better treatment. So coming back to C60, we have used him as a nanocarrier for a drug and the photosynthetizer. Photosynthetizer is the agent of photodynamic therapy for the development of the photodynamic cancer chemotherapy. So in that way, the drug delivery and the photodynamic treatment was combined. And here our cells which were treated with the drugs and not eliminated, and then exposed to the light, and the cell viability was decreased along the all treatment points. The reactive oxygen species in the cells exposed not to the light remained on the control level. However, once exposed to the light, the continuous and very intense increase of the reactive oxygen species production inside the cells was found. That was also followed by ATP drop and K-specific activity increase and translocation of the phosphatidylserine, that altogether give us a ground to conclude that the apoptotic cell death was induced in the cancer cells under treatment of the photoexcited nanocomplexes. The analysis, the statistical analysis of the toxicity give us also the proof of the synergy. So to prove the synergy of the treatment of two effects, the combination index can be calculated that points of whether it's additive or synergetic combination, and if it's less than 1, it points on the very strong synergies. So the combination of one nanocomplex and such light exposure conditions were found the best.
So we continued further also that strategy can be applied for other drugs, as for example, berberine. It's not common, it's alternative drug, it's herbal alkaloid. However, has very interesting anti-cancer activities, but its application is limited by the water solubility and stability. That can be also solved by the synthesis of the nanocomplex. So as an outlook, I would like to point out that from the idea and studies to the application for any nanoparticle, it's a very long story, and it commonly takes 20-15 years from the stage when we could prove that it's effective on the cellular and animal level. However, there is clinical trials, it's very long and requires very high finance resources. But here we have a nice example of the investigation and development of vaccines last years that shows us that it can be done much faster. Hope we can transfer this experience to other fields. So, and this is me. Personally, I love soccer. Actually, maybe it was the primary reason why I moved to Germany. And here I would like to acknowledge our group who is very supportive, and also all colleagues who were helping during the study. My PhD is primary supervisor, the Uwe Ritter from the Technical University of Ilmenau. We would have some more guests from Ilmenau University. And they synthesize C60, they're chemists, and our Ukrainian team. And yes, I for a long time already advertising for our school. So thank you for your attention.
I have a short question, just for information. I'm not from this scientific area, but what does it mean multimodality in your context?
So multimodal. So if we have one therapy, it's monotherapy, it's one modality of treatment. And if we want to combine different therapies, as for example chemotherapy and photodynamic therapy, it would be two modalities. And if we go up, it would be multimodality.
You said about some combinations.
Yes.
Is it a medical problem to combine some types of therapy, or a number of possible combinations are not so large?
Actually, it depends on the chemical structure. Yeah? It depends which nanoparticle, which agents we combine, and which functions they have. So, for example, one agent can have one function, or one agent can have two functions. And it's also then come back. Depends on which cancer, how you would like to approach to treat. You then decide which agent to combine with which. So, but with nanoparticles, this also opportunity of the very high surface to volume ratio gives an opportunity to allow much more molecules in one nanoparticle, so we can combine different agents. But of course, there's the problem of the concentration and the effective dose. So there should be balance.
Do you need a formal approach
to
construct combinations? If you deal with 5 or 10 combinations, it may be not need. But if you deal with a large number of items and combinations with them, maybe it is possible to describe formally which are possible combinations, and then maybe choose one of them or a subset of them. Is it connected?
Yes, it's connected, but nanotechnology is not that far now. We combine like maybe 3 up to 5 modalities with little bit, maybe that we can develop
This is the nearest
more.
future.
Yeah. We work with less number. Yeah? Please, was it us?
Thank you very much for your amazing presentation. And I have a question about your results. It was in vitro or in vivo results?
Yeah, exactly. I presented data which were done in vitro, and our next steps include in vivo. So we do animal studies, but not for this, not in this field.
And other questions about selectivity. Because we use school range just to increase loading capacity, but what about selectivity of destination? So probably we are going to use antibody, yes?
Yeah, so with this example, we rely on passive targeting, so there is selectivity, but if we think how to increase it, we can develop the active targeting that includes antibodies or vitamins, for example. We have one approach where we bound the folate on the C60 as a targeting ligand, for example, but it's yet on the chemical stage, not to the biology. Yeah, of course, you're welcome.
Thank you so much, and it was very interesting for me. I got a lot of new information for me. But I have a question about the result. Which type of answer was reproduced on mouse, yeah, or which mouse?
No, it was a cell, so it's cells grown in the dish. Yeah, and here I presented the results of the leukemic human cells.
And maybe you use combination of two effects of C60, yes, photodynamic and delivery, effect of delivery. Maybe it will depend on the type of cancer disease. Maybe sometimes you need to use some, first of all, maybe some other nanoparticles, or maybe other combinations. It depends on the type of cancer disease.
Yeah. Of course, it's the problem with the cancer because it's a group of disease, and there's so many different types of it, and each one you need to approach specifically. We work also with other cancer models now, primarily, for example, with lung, also cervix and liver cancer. And we develop other nanoparticles as well as polymeric lipids. So, but there also each time it's what to combine depends, of course, of which cancer.
Thank you so much.
A small question, it says 60. You're right, right? Like you don't synthesize it, or
we have partners who synthesize it.
yeah. Okay.
But yeah, you can buy it.
Okay, and my another question. You spoke a lot about drug delivery, but it would be my mistake if I will go away, not asking you. So do I understand right? The C60 comes inside to the cancer cells, not selectivity, but because the cancer grows much quicker than normal tissues, a bigger percentage of C60 or any other drug will get
Nano-drug.
maybe also other, maybe chemotherapy.
Yes, also, but not in that extent. The bigger particle would stay there longer in comparison with this common small molecule.
Bigger because C60 is bigger than normal small drug.
Yeah.
It stays, it stucks in the
Exactly.
cancer cell. And
Not in cancer cell, in cancer tissue, tumor tissue.
And with the time, if we treat with C60 during some time, this level can increase in the cancer tissue, like accumulate inside. And in the end, we have a cancer tissue fulfilled with C60 each cell.
It's our aim.
Yeah, okay, thank you. Very
Yeah.
nice.
Of course, to comment a bit about synthesis there was not that much time to go that deep in details, but I would like to point that goes to the accumulation question, that we work with what is soluble as C60 is lipophilic molecule, you can guess by the structure, and it could be difficult to apply to the biological system. So our partners developed the special synthesis approach to do colloidal water solution of C60. So it means that we don't work with a single molecule of C60, as you imagine a soccer ball, but the aggregates of C60. And so they are 100 nanometer big. And this size accumulates passively better in tumor tissue. And also in tumor tissue, it's two-way. The accumulation is increased, but also the nanoparticles are retained longer in comparison with the normal tissue, because the lymph drainage is also not a bit broken, yeah, it not functions as healthy.
Yeah, thank you a lot.
Yeah, of course.
I have a question concerning the photodynamic therapy. Have you checked in vitro that the additional light increase the cytopathic effect? Yes. And how do you plan to eliminate the tissue inside the body in vivo studies?
Yeah, thank you a lot. It's one of the challenges with photodynamic therapy, as the light has a limit of penetration in the biological tissue. For that way, we develop a few approaches. As the penetration in the biological tissue depends on the wavelength of the light. So if we go to the longer wavelength of the light, it comes better into the tissue. So for the C60, we can use 2-photon excitation. So it means that we need to take double long light that would penetrate deeper. And from other side, we also work, for example, with ultrasound. That high-frequency ultrasound is known to induce the sonoluminescence that is exact in the short wavelength area, right? It's nicely overlaps with C60. So yeah, there are much more to be done.
And we have some more science fiction ideas of using an electron beam.
Yeah.
Okay, we run out of time, and Professor Wojciechowski is already waiting to come to the stage. So the questions